1 minute, ma'am.
Okay.
Yes, 1 minute, ma'am.
Okay, okay.
I'm starting. Yeah, I'm starting
Yeah.
live stream.
Okay. Actually, now it's gone, the screen. Now it's shared. Yeah,
Yeah, just now. Yeah, now it is.
yeah, it is visible.
I'm going to the presentation.
Okay. So, shall I start the class, sir?
Just see how many participants are there. Totally,
Okay,
there
okay.
are minus two.
I think,
I think there are four participants are there.
yeah, I think total of 10 or 8, right? I'm not sure. First time I'm handling this class.
Maybe I think you have informed the classes at around 3:30,
Yeah, yeah,
right?
yeah. Actually, there was some network problem. Actually, the class was at 3 o'clock, but I was not able to start at 3.
Hello.
Hello, can you hear me?
Okay, okay, okay.
Hello, can you hear me? Oh,
Yes, I can hear you.
okay, okay. I think there are 4 participants right now. 4 students have joined right now.
Yes, ma'am. Yeah, ma'am. Totally, there are, I think, two more participants have joined.
Sir, actually, your voice is breaking. I couldn't hear you properly. Hello, hello, sir. Can you hear me?
Yeah, my internet was somewhat not. It is very unstable, ma'am.
Okay, sir. No
Yeah,
problem,
my
sir.
voice, your internet is somewhat unstable in my area.
Yeah. Okay, sir. No
Yes,
problem,
I
sir.
can hear you.
Okay, sir. I think we can start, sir. I
Yes,
think there
ma'am,
are
I can
about
hear you. If you want, I can start the class.
yes, sir. I will ask once the students also, sir. So, hello, hello. So, shall I start the class, students? So, hello. Okay. Then you can unmute yourself. Okay, okay. So, good afternoon to everyone. Myself, Lakshmi, and
Yes, yes.
yes, sir. I'm starting the class. Okay. So, oh, 1 second. So I think sir left. Okay, I think he came back.
No, no, I came. I'm here.
Ah, yes, yes, sir. Yes, yes. Okay.
The screen is visible?
Yeah, yeah. Now it's visible. So
Okay. Shall we start the class now?
yeah, yeah, yeah. So we can start.
Okay. One
Okay,
minute,
sir.
ma'am, before I want to check the live streaming.
Okay, okay,
Okay.
sure.
1 minute. Yeah, the
Sir,
live
actually
stream
the
is,
screen
yeah,
is also
perfect.
gone.
It's going. Yeah,
Actually,
yeah,
the
just
screen is.
intentionally I have removed one.
Okay, okay.
Yeah, now it is out.
Yeah, yeah. Yes, sir.
Now you can start.
Okay, sir. So, good afternoon to one and all. Myself, Lakshmi. I'm doing PhD right now at TNAU Coimbatore. So, today we are going to see on insect nervous system. So, you all know that we are multicellular organisms, but in unicellular organism, a single cell will be performing all the functions of life. So these unicellular organisms, they do not need any specific mechanism for coordination of various activities. But organism like us, that is multicellular animals, their cell size increases and elongate, and it becomes more complex. So a cell, it will turn into tissue, then an organ. So these organs will be having some specific functions. So all this coordination of activities of various organs will become a necessity for the organism, and this nervous system will be useful in all these coordination. So do you have any idea why this nervous system is important for insects? Do you have any idea? Can anyone answer? You can unmute yourself and you can answer. Do you have any idea why this nervous system is important? Akhila, hello. Okay. So for almost everything, this nervous system is important. Like it is a major seat of yes, to transmit information, of course. So it is actually for coordination, learning, memory, intelligence, and also for behavior. Sir, next slide, sir. Yes. So actually, the nervous system, it consists of large number of nerve cells. So this nerve cell is the one which constitute this nervous system. And these nerve cells are what is known as neurons. And these neurons will be derived from the ectoderm while they are in the embryological stage. And this nervous system, they will be having nerve cells. I told that is neuron, and they will be also having non-nervous parts like neurolemma, which will be covering this nerve cells. So first, let's see what this neuron is. So the two most important property of this protoplasm is nothing but the organelle that is inside this cell body. So they are having sensitivity and conductivity. So what is conductivity? Conductivity is nothing but it is the passing of information from one molecule to another molecule. So that is why this nervous system is important for the coordination of all activities in an insect. So as you see here, this neuron, it consists of a cell body. So you can see here it is having a cell body, and this cell body, it is also known as perikaryon or soma. And as in picture, you can see that the cell body is having a nucleus in the center, and they are also having a protoplasmic extension as you see here. So the longest protoplasmic extension that is arising from this perikaryon or cell body is known as axon. So so this side you can see, and also in the cell body this side you can see a shorter ones, and these shorter ones are what is known as dendrites. So generally these axons, as you see here, it gives out a side branch. So this side branch is what is known as collateral. So all these terms are very important. They may ask in the examination. So you have to remember all these terms. So as you see, there is collateral. And also, if you see here, the axon, collaterals, and also dendrites, they are distally divided into fine fibrils. And you can see there, right? A thorn like thing. So that fibrils is what is known as terminal arborizations. So that is what is known as terminal arborizations. So then these neurons, it is actually, as I said, axon, dendrites, and collaterals are the main part of this neuron. Then there comes a glial cells. So each neuron, it is actually covered by one or more glial cells. So what the function of this glial cells? These glial cells will be insulating, and also it will act as a protecting sheath around the neurons. And also there is a nerve sheath. You should not be confused with the glial cells in nerve sheath. These glial cells is the one which will be a protective to the neurons, but the nerve sheath, it will cover the entire or protect the entire nervous system from the hemolymph, and it acts as a separation for this nervous system and the hemolymph. And these nerve sheath, I couldn't find a picture, so I didn't add it here. So actually, these nervous system or nerve sheath, I'm sorry, nerve sheath, it is having two layers. That is, one is outer layer. The outer layer, it will be non-cellular. So it is not any secretive, just it is a protective part. But the inner cellular layer, it is known as perineurium. And this perineurium will be having abundant mitochondria. And this particular perineurium is the one which will secrete the neurolamella. So this neurolamella is being secreted by this perineurium, and it provides mechanical support to the nervous system. Sir, next. So this neurons is being classified based on the structure and also function. So first, let's see how they are classified based on the structure. So as you see here, the first picture, there is a cell body, and they are having only a single process. So if a neuron is having a single process, that is only the axon, then it is what is known as monopolar. And the second one, you can see it is having a cell body and a two process, that is one on the left and one on the right side. So the left side, it is a distal long dendrite, and right side you can see a proximal axon. So they are having two processes. This is what is known as bipolar neuron. And the third picture, you can see that they are having a proximal axon, and this side you can see they are having many distal dendrite processes. So dendrite is the one which decide whether it is bipolar, monopolar, or multipolar. So in multipolar, they are having many distal dendrite and only one axon. So in every cases, there will be only one axon, and dendrite is the one which decides its structure. So next one. So next slide. So next, it is based on the function. So the neurons are, it is classified into three types based on the function. One is sensory neuron, another is motor neuron, and the last one, it is association neurons. So if you see sensory neurons, they are also known as efferent neurons. And these are actually concerned with the perception of the stimuli, and it will conduct them from the site of perception, that is, from a sense organ to the central nervous system. So the sensory neurons are the one which will carry the stimuli from the sensory organ to the central nervous system. And the next one, it is motor neurons. So motor neuron is also known as efferent neuron, that is EFFERENT. So you can remember that is ME. So that is motor means efferent neuron. So these are responsible for conducting the impulse from the central nervous system to the muscles. So sensory is the one which carries impulse from sense organs to the nervous system, whereas motor neurons, it will carry impulse from nervous system to the muscles. And another one, that is association neurons, that is, it will be a link between this sensory and also the motor neurons. So next. Okay. So next it is the synapse. So what is the synapse? Synapse is nothing but it is a space between one neuron and the one axon and the next axon. I will define it. That is the branch terminal of the axon of one neuron. It will come in intimate association with the collateral. So I said what is collateral or terminal urbanization of the another neuron. So the space between two neurons is what is known as synapse. So this synapse, the gap between these synapses will be 100 arms strong. So that is also an important question. This question will also come in ICAR examination. So then these synapse, it is being divided into three types. That is axosomatic synapse, axodendritic synapse, and axonic synapse. So within the name itself, you can identify. That is, if the space is between, or if two connection between the axon and the cell body, then it is known as the axosomatic synapse. So if the synapse is between the axon and dendrite, then it is what is known as axodendritic synapse. And if the space is between the axon and axon, then it is known as axonic synapse. So here what you have to remember is the gap between the synapse, that is 100 Armstrong. So next page. So now let's come to the types of uh, nervous system. So first we studied about the types of neurons. So now we are going to see about the types of nervous system. So there are three types of nervous system. That is central nervous system, visceral nervous system. This visceral nervous system, it is also known as stomatogastric or sympathetic nervous system. And the third, it is peripheral nervous system. And also you should keep in mind that these three are not at all separate. They are actually interconnected between each other. So first, let's see what this central nervous system is. So next. So the central nervous system, it is actually the major portion of the nervous system. And as you see here, they will be having basically they will be having a pair of ganglia in each body segment. So you know that the insect body is segmented, that is they are having head, thorax, and abdomen. So in each body segment, they will be having a pair of ganglia. And this pair of ganglia, it will be connected. It will be connected by a transverse strand, that is horizontal strand. And this horizontal strand is known as commissures. And then a pair of longitudinal strand. So a strand that is connecting the pair of ganglia in a segment. So if a nerve fiber has to connect a ganglion of other, it will be connected through a pair of longitudinal strand, and this longitudinal strand is what is known as connective. So that term also you have to by heart. That is, a transverse strand is known as commissures, and the longitudinal strand is known as connectives. So as I said, they will be having a pair, and the central nervous system itself is divided into three types. That is the brain or cerebral ganglion or supraesophageal ganglion, then subesophageal ganglion, and the ventral nerve cord. So you can see here in the head region. There will be brain and also subesophageal ganglion, and in thorax and abdomen there will be ventral nerve cord. So first, let's see about the brain. So next, please. So as I said, it is actually a congregation of the ganglion. So there are two ganglionic centers in the head itself. That is, one is the brain and another one is the subesophageal ganglion. So this brain, it is actually lying above the esophagus and also in the anterior part of the head. And also, if you see, this brain is the seat of many long-term organized behavior patterns. And as I said before, this brain is actually formed by the joining of three pairs of the ancestral ganglion of first, second, and third head segment. So in morphology, you will be studying the head segment. It is actually divided into 6 segments. So in that, the first 3 segments, it will be joined together to form the brain. And it will be having threefold division. That is, one is forebrain or protocerebrum, then midbrain or deutocerebrum, and hindbrain or tritocerebrum. So first, let's see this forebrain, that is protocerebrum. So as you see here, it is actually formed due to the ganglia of the pre-antennary segment. That is, pre-antennary segment or the first head segment, and it is the one which forms the greater part of the brain. So as you can see here, you can see the mark, I think. So this is actually the bilobe structure, and it is continuous and laterally with the optic lobes. So this protocerebrum is the one which supplies the nerves to the compound eyes and also the oxyli. So the protocerebrum is connected with the visual organs. So the next one, it is deutocerebrum. So this deutocerebrum, it will be lying in the ventral side of the protocerebrum. And this, as I said, this deutocerebrum, it is also known as midbrain. And if you take this, will represent the fused ganglia of the antennary segment. So the protocerebrum, it is the ganglia fusion of the pre-antennary segment, whereas the deutocerebrum, it is the fusion of the antennary segment. And this is the one which will innervate the antenna. So it is also composed of the paired antennary and also the olfactory lobes. So you know that antenna, it is also used to detect the olfactory things also. So this, as I said, this nerves will innervate the antenna, whereas the protocerebrum, it will innervate the compound eyes and oxyli. So next one, it is the hindbrain, or it is tritocerebrum. So this will be lying below and behind the deutocerebrum. So as I said, all these are connected to each other. So this will be lying behind the deutocerebrum, and this one will be formed due to the joining of the ganglia of the third or intercalary segment of the head. And this is also, as you can see here, this is also divided into 2 separate lobes, which are separately attached to the deutocerebrum. And here you can see that the tritocerebral lobe of one side, it is connected with the other side by the commissure. So as I said, commissure is nothing but a longitudinal stand. And this is the one which will innervate the compound eyes and ocelli. That is what you have to remember. And the deutocerebrum, it will innervate antenna, whereas tritocerebrum, it will innervate the labrum. So next, it is subesophageal ganglion. So next slide. So as you can see here, so as I said, the first ganglionic center, it is the brain. So next, the second one, it is the subesophageal ganglion. And this is formed by the fusion of the ganglia of mandibular, maxillary, and labial segment. So I have been using this name ganglia from the beginning. So what is actually this ganglia? Anyone knows? Okay. Ganglia
Group
is nothing but
nerves.
Group of nerves. Correct. So this subesophageal ganglion, as I said, it is formed by the fusion of ganglia of mandibular, maxillary, and also labial segment. So you know that these three are the mouth parts of these three also are the mouth parts of the insect. So this subesophageal ganglion will be lying below the esophagus. So as I said, the brain, it will be lying above the esophagus, and this subesophageal ganglion, it will be lying below the esophagus, and it will be lying posterior to the brain. And this nerve, it will innervate mandibles, maxilla, hypopharynx, labium, and neck. Other than labrum, all the mouth parts it will be innervating. So next, let's move on to the ventral nerve cord. So next slide, sir. So as I said, in the ventral nerve cord, it will be having the ganglia of thoracic and also the abdominal region with their connectives and nerves that will be arising from them. So as you normally there will be three ganglia located in each of the thoracic segment. So there are three thorax segments, you know that prothorax, mesothorax, and metothorax. So in these three thorax there will be ganglia, and in the abdominal region there will be 5 abdominal ganglia in each of the segment. Then and also in abdominal ganglia there will be a contraction also. So each insect or each order will be having different numbers of abdominal ganglia. So as you see here in the picture, that is A. In the picture A, you can see that one or more abdominal ganglia. So you can see the picture 1. In the abdomen, 1 or 2 abdominal ganglia, that is down, 7 and 8. Bottom, you can see there. So it is actually fused with the metathoracic, that is upper way, that is the first abdominal segment. It is being fused with the metathoracic ganglion. So the example will be chironomus, and also in locust also the ganglia will be seen like this only. Then in figure B, you can see that all the 3 thoracic ganglia will be fused together. So it's then the next one, you can see that in diptera and also in hemiptera, all the thoracic and also abdominal ganglia, it will be fused into a single mass which will be located in the thorax. So example will be musa. I mean in most of the diptera and hemiptera it will be like that. Then in D, that is the next one, you can see that the largest number of abdominal ganglia, it will be occurring in larva or adult of this Thysanura. So you can see here they are having 8 abdominal ganglia. And also in male pulex and also many larval forms, they will be having 8 abdominal segment. So ventral, so as I said, first what I said is brain. So brain, it will be in brain there will be 3 types, that is proto, deuto, and trito. So proto, it will be innervating compound eyes and ocelli, whereas the deuto, it will be innervating the hello. Okay. So deuto, it will be innervating the antenna, whereas subesophageal ganglia, it will be innervating the labrum, whereas trito, it will be innervating labrum, whereas subesophageal ganglia, it will be innervating all the other mouth parts. And this ventral nerve cord, it will be innervating the abdominal as well as the thoracic region. So next slide. So next slide. So next is the visceral nervous system. So this is the second type of nervous system. This is actually known as stomatogastric and also sympathetic nervous system. And this particular nervous system, it is actually associated with our alimentary canal. And this nervous system is divided into 3 types, that is esophageal or stomatial sympathetic, ventral sympathetic, and also caudal sympathetic system. So first, let's see what this esophageal or stomatogastric sympathetic is. So as you can see here in the picture down, they are actually having a median frontal ganglion and also a pair of hybrocerebral ganglion and esophageal ganglion, and also a paired ingluvial or stomachic ganglion. So these are the 4 ganglion that this visceral nervous system consists of. And when you see here, the frontal ganglion, it will be situated above the esophagus and also in front of the brain. So you can see the picture. If you see the picture, you will get in clarity. So this frontal ganglion, it is connected to the tritocerebral lobes by a pair of frontal connective. So this connective and commissures are the one which will be connecting the ganglion to each other. So in some insects, if you see the anteriorly, the frontal ganglion will give off a frontal nerves also, and this will be passing through the clypeus, which is also a part of the head. So so here you can see. So as you see here, you can see the frontal ganglion, and from this frontal ganglion, it will be connecting to the hypocerebral ganglion, and this hypocerebral ganglion will go to the corporocardiaca or the esophageal ganglia. So this hypocerebral, it will be connecting to this esophageal ganglion, and then this esophageal ganglion is then connected to the ingluvial or stomachic ganglion that is situated in the posterior end of the foregut. So this whole visceral is being associated with the digestive system or the alimentary canal of the insect. And these nerves from the ingluvial ganglion, it will innervate the neighboring region of the stomatium or the foregut. So next, it is the ventral sympathetic nervous system. So if you see the ventral, the above picture, you can see this ventral sympathetic nervous system. It actually consists of a single median nerve. So you can see the picture. So it is having a single median nerve and also a pair of transverse nerves that is associated with each ganglion. So in each ganglion, it will be having this type of nerve system. Then the next one is the caudal sympathetic nervous system, and this particular system, it will be arising from the posterior ganglion of the abdominal nerve cord, and this caudal sympathetic nervous system is the one which will be innervating the reproductive system and also the posterior part of the gut. So here you can see the visceral, it is being divided into three, that is esophageal, then ventral, and also the caudal sympathetic nervous system. You should know which of the which of the system is innervating which of the nerves. So next it is peripheral nervous system. So here also, as I said, the frontal ganglion, it will be helpful in the swallowing movement, whereas peristaltic movement, that is the movement of the food, it will be controlled by this ingluvial ganglia. So that is also an important point you should remember.
Hello, ma'am.
Yeah.
Ma'am, could you please explain the esophageal nervous system, ma'am? Couldn't understand.
Esophageal?
Yes, ma'am. Yes, ma'am.
Okay. So you can see the picture right there.
Yes, ma'am.
Okay. So this esophageal, it consists of a median frontal ganglion. Hello. So this is a part of the sympathetic nervous system. Okay. You can see the picture right there.
Yes, ma'am. Yes, ma'am.
Okay. So actually, it will be consisting of a median frontal ganglion and a hypocerebral. So these are all the terms that you have to study in the morphological part. So I think Ashok sir will be dealing with this. Okay. So this pharyngeal and all, you know, right? You might have studied the digestive system.
Yes, yes, ma'am.
Okay. So for everything, every movement or for any stimulus, you need a nerve.
Yeah, ma'am.
Yes, no. So this hypocerebral, esophageal, everything will be lying in this sympathetic. So the sympathetic nervous system is the one which will be controlling all this. So, so this sympathetic, it will be having a median frontal ganglion, which will be. So you can see that frontal ganglion over there, above the brain.
Yes, ma'am.
So it will be innervating that frontal ganglion. Then below the brain, you can see the esophageal ganglion and hypocerebral ganglion.
Yes, ma'am.
Okay, so these two will also be innervated by the sympathetic nervous system. So as I said, these are not separated. They all are being interconnected.
Okay, ma'am.
Okay, so
Okay.
so these four, there will be 4 ganglion, and these 4 ganglion will be interconnected, and these all are situated above the esophagus and also in front of the brain.
Okay, ma'am. What are the 4
Frontal
ganglia, ma'am?
ganglion.
Hypocerebral, esophageal.
Hypocerebral, frontal, esophageal, and ingluvial.
Okay, ma'am.
Ingluvial ganglion.
Okay.
Okay. Okay. These all will be connected to the tritocerebral lobes. So before I said what is tritocerebral lobes, right? In brain, you will be having a third region,
Yes,
which will
ma'am.
be innervating the. Okay. That one is the tritocerebral. So these all will be attached to this tritocerebral lobes by a pair of frontal connectives.
Okay, ma'am.
Okay. Okay. And so can I move to the next one?
Yes, ma'am. Thank you.
Okay. So ventral, you understood? There is nothing to understand in that. It will be innervating the ventral nerve cord. Hello?
Yes, ma'am. I got, ma'am.
Okay. The caudal means it will be innervating the reproductive system and also
Yes,
the
ma'am.
posterior part of the gut.
Yes,
Okay.
ma'am.
So now we have studied the 2 nervous system, that is visceral and also the central nervous system. So now there is the third type of nervous system, that is peripheral. So what this peripheral is, this is the nervous system which will be connecting both the central and also the sympathetic system. Hello? Hello?
Yes, ma'am.
Okay. So these peripheral, it will be connecting both these. That is the peculiar character of this particular nervous system. So next. So this Schwann cells. So as I said, the glial cells, it will be covering the, what I said, what is that glial cells?
Protecting, ma'am. Protecting.
What is glial cells?
Those are protecting, ma'am. Glial cells.
It will be protecting which organ? Which cell?
Nerve cell, ma'am.
Yes, nerve cell. So this Schwann cells, it is also a principal glia, and this will be protecting for the peripheral nervous system. Okay. The special name they have given is Schwann cells. So next, next slide, sir. So next we are going to see about how this impulse is conducted in the axons. So as I said, everything should, this insect nervous system is actually a cholinergic system. That is, cholinergic is nothing but it is the use of this neurotransmitter acetylcholine in the transduction of the action potential. So they may ask what type is insect nervous system. This insect nervous system is a cholinergic nervous system. So now let's see how
Ma'am,
this
your
is
voice is not
being
clear.
conducted. So once electrical signal or axionic conduction. Hello? Hello?
Yeah, now
Ma'am,
it is fine,
it's
ma'am. You can
Is it
continue.
breaking?
Yes, ma'am. Can we start
Okay.
from the conduction of nerve impulse and axons from starting?
Yeah. That is, first you will get a signal, that is stimulus, right? So
Ma'am,
after
what
stimulus,
is the
I mean,
type
I
of
will
nerve impulse? What is it, ma'am? Insect. In insect, what is it called?
cholinergic nervous system. Cholinergic system. That is, it will use the neurotransmitter acetylcholine.
Okay.
Okay, for action potential, it will be using the neurotransmitter acetylcholine, and that way this insect nervous system is known as cholinergic system. Okay, this cholinergic is nothing but it is a type of neurotransmitter. I will explain briefly in the next slide. Okay.
Okay.
Okay. So, sir, next slide, sir. Yes. So you all can see the picture, right? So this is the axon or plasma membrane. So you can see here the charges is being conducted. So as you see here, once the nerve impulse along the axons, uh, these nerve impulses will be caused due to the changes in the conductance of this membrane. So in this membrane, they will be having sodium as well as potassium ions. So resting potential is nothing but when there is no stimulus, that is what is known as resting potential. So during this resting state of the axon, the thing is that there will be an excess of sodium ion external to the membrane, and there will be excess of potassium ion in the axon cytoplasm, that is the inner side. So in order to maintain this charge throughout the plasma membrane, there will be a higher concentration of Kl plus ions inside, and also higher concentration of chloride ions outside. So this ion conductance is what makes the uh, plasma membrane equilibrium. So this equilibrium is what is known as Donan equilibrium. That is, a Donan equilibrium is set up across the plasma membrane. Plasma membrane is nothing but the inner portion of this axon. So with the concentration of K plus ions inside and higher concentration of chloride ions outside. So this happens when the axon is in a resting state. So as a result of this equilibrium only, the charge inside of the axon will be negatively charged with respect to the outside. So do you understand? So what will be the charge inside the axon?
Positive.
Inside.
Negative, ma'am?
Negative.
Yeah. Inside it will be negative, and outside only it will be positive, because outside there are more number of sodium, and inside there is K plus. Okay. So now once the signal came, what happens is there will be an conductance. So normally the insect nerves axon, their resting potential will be minus 70 MV. Okay, this is also an important question. So when the insect nerve, it is in a resting potential, the resting potential will be minus 70 MV. And so what is this stimulus? Can anyone define what a stimulus is? Hello. We will be using this word frequently, right? So what the stimulus means?
It's a
Chemical
sudden movement.
response.
Hello.
Yes, ma'am. It's a sudden movement with the help of any enzymes or any.
Okay, sudden movement you are saying. Okay,
Yes.
someone else was also answering, right? What is your opinion?
Chemical response.
Okay, chemical response. So actually both are correct. The actual definition is that any change that brings an alteration in the metabolic activity. So if any changes in the metabolic activity in a living organism, it is what is known as stimulus. Okay. Hello.
Yes, ma'am.
Okay. So as I said, in the resting condition of the membrane, there exists an ionic imbalance between the inside and also the outside, of the axon. So as I said, this is due to the sodium potassium pump. So this sodium potassium pump is having a major role in the conduction of this impulses. So as I said, the Na will be predominant in the outer region of the axon, whereas the potassium ions will be greater in the inside. So if you take how much time it will be more, the K will be more inside is 24 times. That is, the K plus ions will be 24 times greater inside the axon as compared to the outside. It is not that the K plus is not at all present in the outside of the membrane. It is also present, but it will be less. Only in the outside, only in the inside of the axon membrane, the K plus will be more. That is why in the inside it will be having a negative charge. So, the once the electric potential produced, so as I said, it is a resting potential, okay, and also minus 70 MV is the resting potential. So when an impulse, so once now you get a stimulus. Now you get a stimulus, and when this impulse reaches a point on the membrane, the permeability to the sodium will increase tremendously, resulting in an inward movement of the sodium and also an outward flow of the K plus. So now the sodium, it will be moving inside the axon, and the K plus, it will be moving outside the axon. So this leads to a potential charge inside the membrane. So as I said, there will be negative charge inside and positive charge outside. But once the voltage pump, it is being opened, here the sodium will be the permeability of the sodium will be more, and sodium will be entering inside the axon, and there will be positive charge inside. And now the volt will be 80 to 100. And at this stage, the adjacent areas. So the adjacent areas. So you can see here, some of the areas are positive, some of the areas are negative, and some of the areas are positive. So only where the impulse reaches that area only will be changing its conductance. The other areas where the impulse has not reached will be in the same resting potential only. So now when it reaches, as I said, inside it will be positive, outside it will be negative. So you can, from this we can state that a current flows will be in a local circuit that is away from the point of depolarization. Depolarization is nothing but when an impulse reaches a part of the axon, the particular part, it will become negatively charged outside and positively charged inside. So when this current reaches an area of resting membrane, it will be producing a depolarization. So I think you understood this one. 1 second. Ma'am. Hello. Okay. So now you know what an action potential is. You know what an resting potential is. So now the potential has reached the presynaptic place. So now the impulse has to reach the synaptic one. So this synaptic conduction will be a biochemical conduction. So as I said, what is synapse? Hello.
Hello, ma'am.
What is the synapse?
The gap between the two dendrites or axon or neuron or two different neurons.
Okay, what is that? I mean, how much will be the gap?
100 angstrom units.
Yeah, 100 Armstrong units. Okay. So now the electric or nerve action potential, it arrived the axon terminal. And when it reaches the, next slide, sir. Okay. So as I said, once the electrical or nerve action potential at the axon terminal, it will causes the liberation of the acetylcholine. So as I said, this insect nervous system is a cholinergic system. So it will be using this acetylcholine to the transduction of the action potential. So once this acetylcholine is liberated, certain mechanism will take place. So first, let's see this where this acetylcholine will be present. So you can see here in the picture a particular receptor, I mean vesicle, the circular portion. It is actually known as the presynaptic vesicles. So this presynaptic vesicles, they will be having the acetylcholine, and this acetylcholine will be positively charged. So once the axionic conduction, it reaches the presynapse. That is, once the action potential, acetylcholine, it reaches the axionic conduction. Once it reaches the presynapse, the acetylcholine, which is in a inactivated state, will be getting activated. And after this activation, this acetylcholine, the vesicle will open, and this will move towards the post-synapse stage. And there it will be liberated with the movement of the nerve impulse. So first I said the area, it will become negatively charged, in negative first in resting potential. Inside it will be negative, outside it will be positive. So once the impulse reaches the ion conduction, it will change. So inside it will be positive, and outside it will be negative. So once this negative charge, or the negative charge reaches the presynaptic portion, the presynaptic vesicle, it will be having acetylcholine. So this once this acetylcholine is being activated, this vesicle is being activated, the acetylcholine is being liberated. And once the acetylcholine is liberated, it will be combined with the receptor protein. So the receptor protein, it will be combining with the acetylcholine, and also there is another ion, that is calcium ion, which is present in the vesicle outside, will be activated. Only when this calcium ion when the calcium pump is open, and when it reaches the vesicle, only this vesicle will open. So for the vesicle to open, the calcium ion is very much necessary. Only if the calcium ions activate the presynaptic vesicle, the vesicle will open and the acetylcholine will be liberated. And this presynaptic depolarization, this will increase the permeability of the membrane to calcium. So once the negative charge inside the axon reaches the presynaptic area, the permeability of the calcium will also increases. And after the permeability of the calcium, it will enter into the presynaptic vesicle, and this presynaptic vesicle, it will open, and the acetylcholine, it will be reaching the synaptic place. So once the calcium ion, it diffuse into the presynaptic terminal, what happens, as I said, the vesicle will open, and the acetylcholine will show its activity. So once the impulse, it has been triggered at the postsynaptic region, what happens here is, that is, once the acetylcholine reaches the synaptic region, the acetylcholine, it will be broken down into acetic acid and also choline. And this activity is actually done by an enzyme called acetylcholine esterase. And this acetylcholine is actually again resynthesized in the presynaptic area in the presence of another enzyme, that is choline acetylase. Hello? Hello?
Yes, ma'am.
So you understood this point? Actually, I was stammering at this time. Hello?
Yes, ma'am.
You understand the mechanism of this one, or you wanted me to explain it once again?
Ma'am, explain once again. Now it's too confusing.
Yes,
Okay.
ma'am.
So from first itself, I have to explain.
Up to depolarization, it's okay, ma'am.
Okay. In the axionic conduction, it is okay. Hello?
Yes, ma'am. Yes.
So up to axionic conduction, it is okay. You need synaptic conduction.
Yes, ma'am.
Okay. So as I said, once this action potential, this axionic conduction, when it reaches the presynaptic area, okay, there will be having vesicles.
Okay, ma'am.
Okay. So when this conduction reaches the presynaptic vesicle, what happens is the calcium pump, which is present, also will be opened.
Ma'am, calcium
Okay.
pump is outside the synapse or inside the axon, ma'am?
It is actually present outside the synapse.
Okay.
Okay.
Yes,
It
ma'am.
will be in the axionic, nearby the axionic. It
Okay.
is not in the synapse. This is all happening inside. That is, I said no, presynaptic.
Okay, ma'am.
I didn't come to the synaptic area. Now it is like synaptic presynaptic only.
Okay,
So
ma'am.
these all will be taking place inside the axon, just before the synapse. Okay,
Okay, ma'am.
you're getting it?
Yes, ma'am. Screen.
Okay. Okay. So as you see here, this vesicle, no, you can see here, right? The synaptic vesicles.
Yes, ma'am.
So it is actually inside the axon, not outside it.
Okay.
So only okay. So this acetylcholine is actually present inside the axon only. At the end of the axon, the acetylcholine will be present inside the vesicle. Okay.
Okay, ma'am.
So once this axon conduction comes to this presynaptic area, what happens is inside you can see a green color pump
Yes, ma'am.
that will be opened. So this will allow the calcium to enter inside the axon. Okay, and this calcium, it will be the vesicle will be permeable to this calcium.
Okay.
So once this calcium enters into the vesicle, what happens is the acetylcholine, it will be activated, and it will be joining with another docking protein.
Okay.
And this acetylcholine will be released into the synapse now. Or before all these activities are actually happening inside the axon.
Okay, ma'am.
Okay, so once the vesicle opens, the acetylcholines will be reaching the synaptic area.
Okay, ma'am.
Okay, so once it reaches,
The synaptic conduction starts when there is negative charge outside of the axon, ma'am, or inside, ma'am?
no, that's what I'm saying. Once the axionic conduction reaches, that is, axion conduction means what? Inside it will be negative only. No, during the depolarization state only it is happening.
It is outside negative charge, ma'am.
Actually, the axionic conduction, it will stop when it comes to the presynaptic area.
Okay.
Okay, that is when this axionic conduction, it reaches the vesicle, it will be having negative charge inside. I mean outside, and positive charge inside.
Yes, ma'am. Yes.
Okay. So now the acetylcholine is being released in the synapse. Okay.
Yes, ma'am.
And once this is released, there will be receptors in the postsynaptic area. This postsynaptic area will be in the next neuron. Okay. This will receive this acetylcholine impulses, and the impulse will be conducted again and again.
Okay, ma'am.
Okay, that's what. And also, as I said, this acetylcholine, it will be broken down because it has to be synthesized again and again. No.
Yes, ma'am. Yes.
Otherwise, they will become exhausted. So so there is an enzyme called acetylcholine esterase. This particular enzyme, it will break this acetylcholine into two, that is acetic acid and choline. Okay, then this acetic acid and choline, it will be again joined together by another enzyme called choline acetylase to form the acetylcholine.
Okay, ma'am.
You understood? So these are the two enzymes that is showing this type of activities.
Okay, ma'am.
So this is how the impulse is being conducted. So potassium, calcium, and also sodium. It is having these pumps are having a major role in the conduction.
Okay, ma'am.
Okay, you understood now?
Yes,
Axonic
ma'am.
and synaptic conduction.
Yes, ma'am.
Oh, sir, next slides. So this is the reaction that takes place. That is the acetylcholine, A choline. It is being that is it will use the energy, and the choline esterase, it will form the acetylcholine, and then again the acetylcholine esterase, it will be degrading the acetylcholine into acetic acid and choline. So this is the formula you should by heart. So next slides. So this is the phases. That is nothing. First I said it is the resting potential. Resting potential is nothing but inside it will be negative charge and outside it will be positive charge. Then it is the rising phase. In rising phase there will be that is the conductance of the permeability will start. That is inside it will become positive and outside it will be negative. Then again after this then again there will be falling phase. That is the signal's effective will be reducing. So next, so there will be three phases. That is resting potential. First is resting potential, then there will be rising phase, then there will be falling phase. Okay, so here you can, yes. So as I said, after the rising phase, here the permeability of the membrane will be increasing to potassium, which will be resulting in a greater influx of this potassium, and the charge of the inside membrane will be negative. So for, as I said first itself, and this will constitute the falling phase of the action potential. So due to the greater inflow of K plus, that is the action potential, that is spike, the peak will be only for 1 or 2 milliseconds only. So after that what happens is the inflow of K plus will become greater, and this will result in the positive phase. That is inside the K plus ions will be more. But now also outside also there is accumulation of the K plus. Okay, so because of this accumulation of the K plus outside, there will be negative active potential. So after this negative after potential, the Na pump, the Na will be moving outside. So now there will be normal stage. So first there will be resting potential, then there will be rising phase, that is inside it will be positive, outside there will be negative. Then slowly it will reduce, that is inside it will be becoming negative and outside it will be becoming positive. Then later what happens is so the K plus permeability will more, the Na plus permeability will less. So after this negative after potential, what happens is the Na will also started moving moving outside the axon membrane, and that is what is resting potential again. So next page, sir. So now let's see some of the some other important terms that you should know in this nervous system. So it is axon hillock. It is a term nothing but when an axon it arises from the part of the nerve cell, that is known as axon hillock. And the next one, it is neuropile. Neuropile is nothing but the mass or group of axon. So we said that a mass or group of nerve cell is ganglia, and mass or group of axon is neuropile. And do you have any idea what will be the normal diameter of this axon?
No, ma'am.
Okay, it will be just 5 micrometer, and that is the normal diameter. But there are axons which will be 60 diameter inside the insect body, and this axon is what is known as giant fibers. Hello.
Yes,
Okay.
ma'am.
That is okay. That is what is known as giant fibers. And neurilemma, it is nothing but the ganglionic surroundings. So the ganglion, it will be surrounded by this neurilemma, I already said, and neurolemma is nothing but the axionic surrounding. So only a single word is being changed. So I, it comes for ganglionic, and O, it comes for axionic surroundings.
Ma'am, axon hillock means what, ma'am?
Which one? Which one?
First one, ma'am.
Axon-
Axon hillock.
Axonic surrounding. Just a protective sheath of axon is what is known as neurolemma.
No, ma'am. Axon hillock. First one, ma'am.
Axon hillock is nothing. Just an axon that arrives from a nerve cell.
Okay.
That is the axon hillock. So, as I said, the insect, it is actually, as I said, I said no, it is cholinergic system. That is, it uses the acetylcholine for the transduction of the action potential. But beside acetylcholine, there are also many neurotransmitter that is present in this insect, like dopamine, octapamine, histamine. These all are neurotransmitter, but this doesn't have any role in transduction, but they are having roles in molting and all. Okay. So next it is so that's all. And so next. So do you have? So anyone knows which of the animal is not having nervous system? It is beyond insect. That is, it is not insect, but any other animals or multicellular organism which is not having nervous system. Hello.
No, ma'am.
You know sponges. Hello. Hello.
Yes, ma'am.
Actually, I was audible before. Hello. Hello, sir.
Oh, now it is audible, ma'am, but previous it is not.
Okay, it was not. Okay. I just asked a question that which of the animal is not having nervous system? Anyone have idea?
No, ma'am.
Okay, actually, jellyfish, it is not having nervous system. Okay. Hello.
Okay, ma'am.
Okay. Thank you so much. Actually, I was busy with my research work. That's why I was not able to take class yesterday also. So if you have any doubts, you can contact me, and you can ask even now also. I will put up my phone number in the group. Okay. So if you need any materials or if you have any doubts, you can ask me. If you have any feedback also, you can.
Will we get the notes of this class?
Yeah. Shall I prepare and give you within 2 days? Because I was
Okay.
little busy with my research work. Surely I will send you before. Okay.
Okay, ma'am.
Before
Thank you.
next class, I will be sending you the notes. I will prepare it for you and I will send. Okay. Because it is splitted in all the books, so I will. I will coalesce it and I will send you.
Okay, thank you, ma'am.
Okay. Okay. So next class I will be taking on excretory system. So I will be taking on Friday, I think. Okay. Before that, Ashok sir will be dealing with you regarding the morphological part.
Okay, ma'am.
Okay. You all are from which year actually?
Ma'am, I'm from third year.
Third year. Okay. Okay. Fine. So you know somewhat about morphology and all, right? Whatever the terms I was using, you are familiar with, right?
Yes, ma'am.
Okay. Okay. All are from third year only. Anyone who is from second year or
I'm from final year, ma'am.
yeah, from final year? Okay.
Yes, ma'am.
Actually, how many students are there of total?
In our WhatsApp group, there are nearly 12 to 13 members, ma'am. Actually, we don't know, ma'am.
12 to 13. Fine, fine. So if you have any to be added or you need any question banks, you can ask me, okay? I will help
Okay.
you at any time. You can contact me. I will put up my number in your group. Okay?
Ma'am, can we contact through WhatsApp, ma'am?
What? I didn't get you.
Contact you
Yeah,
through
yeah,
WhatsApp?
you can.
Okay, ma'am.
Yeah, you can contact me at any time through WhatsApp or call. Anything is fine. Okay?
Okay, ma'am.
Okay. Shall I conclude the class?
Yes, ma'am. Thank you. Interesting class, ma'am.
Okay, thank you so much. No one is having doubt now. I will try to collect more information because it was really very tight for me these days. That's why I couldn't handle more classes. I will try to make it more interesting for you, okay, in the upcoming classes.
Okay, ma'am. Thank you.
Okay. Thank you so much. If you have any doubt, you can contact me on WhatsApp. Thank you so much. And thank you, Aditya, sir,
Thank
for
you. Thank
helping
you.
me out. Okay, sir. Thank you.
Okay, students, thank you very much.
Thank you, sir.